Chris, is there feedback? Can you turn down the volume a little bit, perhaps? Or maybe it's that one. Thanks a lot for coming. Thanks, Mafalda, Johnny, Benedetta, Ben, for organizing this wonderful meeting in this beautiful place. And thanks all for coming. It's been a pleasure so far, and yeah, it's a really nice meeting and community. So today, I would like to talk to you about our work on thinking about the effects of protein folding, degradation, stability on missense variants. Most of what I'll talk about is a close collaboration with Rasmus Hartmann-Petersen, who's also here. And we both have a shared interest in protein folding or misfolding and degradation. And so that's really how we entered this field. And so that's sort of the theme, although I will give you some other side stories as we move along. And in particular, I'd like to, at least towards the end, tell you sort of something about differences between protein stability and protein abundance and protein degradation, that these things are highly related but not quite exactly the same, and once you take into the protein quality control apparatus. But I've come to realize that at this meeting, and I actually think that that's an amazing thing, right, that we start at the end of the patients. And so I'd like to start with a very brief entry into a sort of a fun but interesting story about missense variants in a gene called glucokinase that our colleagues in Copenhagen, Torben Hansen and Nette Giesing, was working on. And it's sort of an interesting story in the sense that the missense variants turn out to be good. So I'll just walk you briefly through that. So Torben and Annette had selected a group of people that had just been diagnosed with type 2 diabetes after taking an oral glucose test, and then they sequenced them. So these are patients go to the doctor, you drink glucose, and then you measure how much glucose you have in the blood afterwards. And then what they did is that then they sequenced, and then they found a bunch of a small fraction, about a couple of percent of the patients that had been diagnosed had what would be assigned as pathogenic variants in glucokinase. And then they characterized the phenotypes of these people. And so this is shown here in these slots with 3 different phenotypes of sort of a controlled non-diabetic group, and then people that were assigned as diabetics, but those that are either non-carriers or those that are carriers. And so it actually turns out that people who respond in the wrong way, so to speak, to an oral glucose test, but have glucokinase variants, actually behave phenotypically, apart from the glucose test, more or less like control variants. So they have a slight misadjustment of their blood glucose level, but apart from that, they're actually perfectly fine, and you should just take them off treatment. So this is a nice example, I think, of where being able to characterize these missense variants would actually just basically cure people because they're fine, right? They really don't need much test, apart from the fact if they get pregnant and other things, and this is really outside of my realm. But of course, as in all other genes, there's lots of variants of uncertain significance. So so Sarah from Rasmus's group made a beautiful map of glucokinase by yeast complementation. There's lots of variants that cause loss of function. There's quite a few variants that cause gain of function, and so this is published. I'm not really going to go into lots of details about this, just to say again, we and in this case, this is really Sarah's work, can measure variance at scale. I just wanted to show this data just because I think it's so nice, and it's not my data, but you can correlate the MAVE data that you measure by yeast complementation with catalytic efficiency, purified proteins. You get a nice correlation both for increased function and decreased function. You can even correlate the yeast-based data with fasting glucose levels in individual patients. It's clearly not a perfect correlation, but given biology knowledge of human beings, I think that this is pretty impressive. And then finally, this is just to say there's actually 2 different diseases associated with missense variant and glucokinase. So again, in all of this discussion about gene-specific cutoffs, right? And this, we in some cases, of course, need disease-specific cutoffs, right? That there is a disease that's loss of function, and there's actually another disease that's increased function of glucokinase. And so we would ideally need to be able to have that. And it's clearly not a perfect separation from the very few benign variants that are known, but at least there's a reasonably good AUC for these 2 groups of patients. So, glucokinase, a fascinating enzyme. I won't have time to talk too much about it, only to say that we are very interested in understanding also the mechanisms of these things. And we've done quite a bit in this paper, but what I'll talk about now is again Sarah's work on trying to deconvolute the effects of protein abundance from changes in intrinsic activity. And the way that we do this, and we've heard examples of this previously, is that we sort of do independent measurements of the abundance of the variants and then the activity as proved in this case by yeast complementation, as I just showed you before. And then I'll move on to later to show that we can try to do something similarly computationally. So I'll get back to that in a second. So this Sarah repeated, her assay, in this case measuring the abundance, and this is sort of the 2D map of the activity scores and the abundance scores, and there's a bunch of variants that we sort of, with these cutoffs, classify as being sort of wild type like, and then there's a bunch of variants that are loss of function, and we find in this case that about 40% of the loss of function variants can be explained simply by the fact that their abundance is very low. And this is a number that's comparable to other things we've seen at this meeting, and it's roughly the number, as I'll show you in a second, that holds true for variants across the genome. This is actually a super interesting enzyme, right? For those of you who have studied metabolism at some point, glucokinase is this weird enzyme that's a little bit crippled, which is why you can actually relatively easily get gain of function. And so there's multiple states, so some of this variation we can actually explain by shifting between sort of active and inactive conformations. But this is all described in the papers. Before this work, we'd actually done work taking data from the literature on proteins for which there had been these kind of measurements where the people had independently measured activity and abundance. There's data from Doug's lab and from several others. I'm showing you here, actually these are both from a mixture of labs. I'm showing you here sort of a similar kind of plot, and I apologize. While we make a lot of these plots, we have actually decided always on what is on what axis. So down here are now the variants that are blue, which are the ones that have high abundance but low activity. Up here again are the wild type like, and here the ones that are sort of loss of abundance and loss of function. And again, this is work from Matteo analyzing these things, and again showing that in these cases there's a little bit more than 50% of the variants that cause loss of function together with loss of abundance. So although our main focus was originally at least on understanding these variants down here, we took sort of a little side tour to try to look at the other ones. What are these variants where you lose activity without losing abundance? And when I'm saying this, it sounds sort of obvious, right? That these would be variants that hit important functional sites. Right? And so this is zooming in on not T15's active site and the active site of P10. And all these blue residues are residues where most of the variants cause loss of activity as measured by the MAVE, but not loss of abundance also measured by the MAVE. And so it's a very convenient way of finding residues that are important simply because if you mutate them, right, they lose function, and they don't lose it for this kind of trivial reason that the protein is gone. So it's a very convenient way of finding residues, not just in the active site, but also surrounding the active sites. We find actually quite a few number of sort of second shell variants and even things on the back of the proteins that change activity as measured by whatever the MAVE is measuring without really substantially decreasing abundance. So this is data we have for maybe 3 or 4 different proteins now, maybe a little bit more. But of course, we'd like to do these kind of analyses at scale. And so we switched to computational modeling, and it was maybe not sort of a super ingenious thought that we say, well, maybe we can predict these things independently of each other. And so we ran predictions that were either based on predicting changes in thermodynamic stability or measuring, how likely a variation is based on, some conservation model. We've used many different models, ranging from the Gemma model that you just heard to, a number of other models. And what we find, this is just for one of these proteins often, is that, the stability calculations tend to correlate more strongly with abundance measurements than the conservation scores, while you get the reverse when you're measuring something related to function. And again, that makes sense, right? Stability and abundance sort of correlated. What evolution cares about is really activity, whether changes in activity is due to changes in abundance or hitting the active sites. So what we thought was that we can use these predictions now as proxies for sort of stability or abundance effects or functional effects. And we can then use these to deconvolute the effects. And so Matteo played around with this, and there's a couple of papers published, but we published this paper a while ago where we took these simple features. We calculate the effect of all mutations in a given protein with stability and with conservation, and then we say, well, there's these residues. They are obviously important because evolution cares about them, but they're not important for stability reasons because the change in thermodynamic stability is very small. Maybe we can use that as a simple classifier for finding functionally important sites in proteins. And so we trained a tiny little model, mostly taking these kinds of conservation and stability scores into account. We trained it against the MAVE data that was around. It's a relatively small number of data points, or small number of proteins, so we made a very, very simple model, but it turns out actually worked relatively well. And I'm skipping most of the validation. You can read that in the paper, but we can now do things like take a random protein and find the residues that are important either for stability or important for function. And so here's just one example, just to show you the kind of information we can get out. As you know, blue residues important for function, but not due to stability effects, cluster around active sites or protein-protein interfaces, or with sort of co-regulators. So it's a very simple, not perfect, 70–80% kind of accuracy approach, where we can not just score variants whether they are good or bad, but we can also sort of put them at least in two separate groups. Are they good, are they bad because they hit some important site, or are they bad simply because they screw up the protein structure? So this enabled us then to scale up these analyses. To do that, we had to switch to something that was slightly faster than the kind of Rosetta calculations that we were doing. So we switched now to using various types of language models. And so we have this preprint out now where we basically did this same kind of analysis on the entire human proteome using ESM-IF as a proxy for protein structure and using ESM-1B as a way of scoring whether evolution cares about some specific residue. So not so complicated. Take all the structures from the AlphaFold database, validate that we can do these calculations using predicted structures, run them through, and then basically use ESM-1B as a classifier of loss of function, and then use ESM-IF on the loss of function variants, whether they're bad for stability reasons or bad for other reasons. Oops. So this is, again, we played around with this, doing this. We ended up doing it in the most simple possible way using simple cutoffs. So we basically find a cutoff of the conservation scores using protein gym, so have some cutoff of the ESM scores, and we use this mega-scaled protein stability dataset to find a cutoff for the ESM-IF scores. So a very simple model, simply are you above or below some score, and use the same scores for all proteins with all the problems that come from that. So again, just to return to glucokinase, just to show you how this works. This is the experimental classification. Blue means residues that cause loss of activity in Sarah's MAVE, but not loss of abundance in Sarah's MAVE. Red means residues that cause loss of activity and abundance, and green are sort of the rest. This is our two models, the original model and the new model. You can see here, for example, in the active site, we don't have a perfect classification, but we have a relatively good classification, in particular given that these kind of sites is a relatively small factor. Then there's some statistics over here on this protein, and we've compared against a bunch of other things, and this is all described in the papers. So again, just to validate that the model is not completely crazy. So what does it look like if we look at things across the proteome? So with the cutoff that we choose, we have a relatively good classification of benign variants and a relatively good, but clearly not perfect, pathogenic variants. We have about 20% misclassification of variants that we predict to be wild-type-like, but that are really, classified as pathogenic, and that's of course the cutoffs that we've chosen, in particular for the conservation score. We could have pushed this around, but this is sort of what we ended up with. But if you sort of ignore that for a second and look at these variants over here, we find that a little bit more than half of the variants that we classify as loss of function are due to loss of abundance, and then a little bit fewer are because they hit important active sites. Right? So again, in line with other people have suggested, loss of abundance is clearly the single most dominant mechanism for missense variants across the proteome. We can now start to break that down, looking at different proteins, different types of the structure. Maybe not so surprising, but gratifying to see. If you, for example, look at buried residues, you have a very large contribution, of course, from residues that are important for protein stability. And if you look at solvent-exposed residues in folded domains, you have the opposite, right? You have an enrichment in what we call functionally important residues, catalytic residues and enzymes, protein-protein interactions. We can also see here in disordered proteins, obviously protein stability is not, at least in the structural stability, not an important mechanism. So we can now take these kind of data and start to sort of dig into things and sort of do mechanistic studies of individual proteins. And of course, you can just all look at the data. It's available online. So until now, I've sort of made the symmetry between protein stability and protein abundance, but as many of you will know, these are actually not the same thing. They're related, but abundance is clearly something else. And so we've started sort of to peel away at this, and I'll show you some data. It's all pre-printed, and it's not all, some of it is very clear, but some of it is not so clear, but we're still trying to figure out how best to analyze these. In particular, we're interested in trying to figure out how we can take information about the protein quality control apparatus into account. And so some of the work is done by Thea, who's also here, that tried to sort of look at the VAMSIC data from 6 different proteins, from Doug's lab, from Rasmus's lab, and from a couple of other places, and sort of try to look at it, across the scale. And the first thing Thea did was actually very, very simple. Can we come up with a simple null model for explaining measured abundance data? And so what she did was very simple. She took proteins, she took one protein out that we wanted to predict, and then we create just substitution matrices from the VAMSIC data from the other proteins and see, can we just take, is a residue exposed or buried? And then we look up, the effect of a valine to leucine or tryptophan to arginine, averaging over, site independently from all the others. And that actually works pretty well. It doesn't work so well if you just train such a model on a single MAVE dataset, but as you increase average over more and more and try to predict something that you didn't include in, actually you sort of seem to be, converging on numbers that are certainly competitive with much fancier models of abundance. So we can explain most of the measured VAMSIC data by simply looking up in a substitution matrix and using a different substitution matrix, whether the residue is solvent exposed or buried. But you can also see, and this is something maybe we don't talk so much about, right, that these correlation coefficients are not so high. So there's certainly lots of stuff in there that we are not capturing with this model, but with all other models. And so we started asking, why is it so difficult to learn across these MAVEs? And this is now the score distribution from these proteins, and these are sort of the average scores. And you can see that they're very different. And we don't think that this is because these proteins are very different fundamentally. It's something that is related to the way that the experiments are done, how you do your sorting, how you create your library, all these kind of things, affect the distributions, in addition, of course, to protein-specific effects. But these are all nice sort of globular proteins, and all the experiments are done very carefully. And remember, these are all exactly the same experiment, right? Done in the same way, site saturation library, fact sort into 4 bins, take them out, sequence them, and processed in the same way. And so one reason why the difference is shown in this plot, which when I first saw it surprised me somewhat. So this is, again, data just collected from the literature, again, from various people's labs. And what I'm showing you here, all these points here, are correlations between low throughput measurements and high throughput measurements. And in all cases, there's a nice high correlation, but it's not all linear, and it's not always non-linear in the same way. So here's a really nice example. Again, this is from Doug's lab, where there's a nice linear correlation between the low throughput abundance measured one by one and the scores you get out from a MAVE. Here's another case where it's clearly non-linear, where a VAMSIC score of 0 can be either 0 abundance or sort of halfway to wild type. Right? So the same score can really mean quite different things in terms of actual abundance. The same over here. Here's another case where it's sort of the opposite, where low abundance can really get sort of a relatively wide range of VAMSIC scores. And so this is not a problem if you're just looking at a single protein and you're looking at a rank correlation, But if you're trying to train a model across all of these data sets, and you have these large-scale data sets on the y-axis, it's sort of tricky to compare the scores across different proteins, at least if we do it with these sort of processed MAP-seq scores. And so we've done a few different things. I'll just show you very briefly one thing that Tia has done, which is to train a model against the full data set, but taking this sort of experimental variation into account. And she had a poster on it the other day. So the approach is relatively straightforward after we figured out how to do it. And that is we train a neural network that uses ESMIF as input that predicts abundance in the sort of the physical scale, right? So the low throughput kind of scale, the numbers that we care about. But we only have on the order of a few hundred data points. So then we run these predictions through these kind of sort of standard curves, and then we can predict now, using these standard curves on the much larger data set, and then we can create a loss function against this larger data set. So we can train a model that predicts abundance, and then we can transform these numbers into sort of a more experimentally relevant scale. And we've done a lot of these things and played around with doing it in many different ways, and it turns out to work, but actually the improvements are relatively modest, telling us that maybe this isn't the final solution, but this is something that I think people would be great to think about. I'm showing you one case where there is an improvement. So this is here on ASPER. So this is the mean absolute error, and this is the correlation coefficient. This is if we don't take these standard curves into account, we get a high mean absolute error, but we sort of really decrease the mean absolute error against the MAP-seq data and concomitantly get an increase in the correlation coefficient if we take this experiment-to-experiment variation into account. And on average, it improves things, but the improvements are relatively modest. So I think that the conclusions of this are at least twofold. One is it's really great if people measure something more directly related to the phenotype on a subset of variants and deposit it together with the wave data, so we know exactly what the molecular phenotype is. That enables us to do these kind of things. And secondly, we probably need other approaches for dealing with things. And Tim showed something nice earlier, right, where that they've been working on for many years, where you have variables that you can tune, like concentrations, and of course for mutations, you can oversample the mutation space. We've done that, Ben Lehner and others have done this. And so that, of course, sometimes helps. But in general, it would be really nice if there is sort of a more systematic way of really knowing exactly the molecular phenotype that you are testing. So in the last time, I would like to tell you about another side of abundance that isn't related directly, at least, to protein stability, or at least not only related to protein stability. The data is also all published or pre-printed, so I'll just give you a few sort of snippets of what we found. Here's just one example that I'll sort of lead within. So this is, again, Rasmus Lene's MAP-seq data of Parkin, and this is the structure of Parkin. It has the usual kind of structure with lots of stripes here, the folded domains, and a stripe here along proline. There's this long disordered linker that is functionally important. And then you can see up here, there's a bunch of variants in this disordered linker that really causes low abundance. And if you can't probably see this, but if you zoom in, these are all insertions of hydrophobic amino acids. And this is in a region that is already a little bit hydrophobic, and this you can maybe see with these yellow wild types here. So if you have a stretch of residues that's borderline hydrophobic for functional reasons in this case, and you put in an extra hydrophobic, you create a little hydrophobic patch. And this hydrophobic patch is then recognized by the protein quality control apparatus as something that looks like something that used to be buried inside the core of a protein but is unfolded. And then the quality control apparatus says, let's get rid of this. And so these proteins are degraded simply from the folded state, but because they're exposing a hydrophobic Degron motif. So this one can actually test, and again, this is Rasmus and his lab's data. You can now tile this protein, take these tiles, stick them onto the same reporter protein, make variants, and what this data shows that if you take the tiles over just this loop and you introduce the variants into those tiles, you can basically recover the abundance of the full-length protein simply by looking at these tiles, right? Showing that the reasons why these variants are low abundance is not because of the rest of the protein is doing something, but it's just that there's a local sequence motif that is being recognized by the quality control apparatus and causing degradation. So these are these two tiles up here in this protein that have been tiled, that have then a bunch of mutants, and you can see now a nice correlation between the full-length abundance scores and the abundance scores of the tiles. So this would be, I think, nice to know at scale. And so again, Rasmus's lab, and this is work mostly by Vasilius and Fia, and Doug was involved in this also, did sort of a similar experiment, tiling either all cytosolic proteins and putting them in the cytoplasm, or tiling all human transcription factors and targeting the nucleus, and then doing this kind of sort-seek experiment and measuring the abundance of these. So the data is again all out now, so I'll just give you a few snippets. There are differences between being in the cytoplasm and the nucleus. I'm not going to tell you about that, but that's described in the paper. They're relatively small, so most things behave similarly whether you are in the cytoplasm or the nucleus. The peptides that are low abundance can be restabilized by either inhibiting an E1 ligase or the proteasome, so they are degraded by the normal ubiquitin proteasome quality control system. So that's sort of nice, right? It's not aggregation or something else. It's really degradation, and that's the reason why they're low abundance. If we just average across all the data, this is the kind of map that you get. So you see that the effect of all the amino acids is more or less position independent across the entire tile, apart from the C-terminus. So we took these things and stuck them at the C-terminus, so there's a fluorescent protein towards the N-terminus, and there's a few variations across the C-terminus. So if you subtract off the average map, this is what you get, and then you recover known C-terminal degrons, a single or maybe stabilons, so something that improves abundance on average, plus maybe some new degrons. And there's a little bit of difference between these degron motifs, whether you are in the cytoplasm or whether you are in the nucleus. So for example, if your protein ends in GG or EE or something like that, you will have a lower abundance than you would, if you ended with methionine. And there was a nice data set shown yesterday where there were stop codons towards the end that increased abundance, and that protein ends with a C-terminal degron. Right? So I think that there will be lots of these things that people will see, and some of this is known and some of this is new. So now we can take these tiles and we can then go back and look at full-length proteins. Again, this is Rasmus and Vasilius' data. So here are two proteins where there are tiles that are strong degrons, and if you look at the abundance of the full-length protein, they're relatively low. They can be stabilized by inhibiting the proteasome, but if you remove the degron again, you increase the abundance of the protein, showing that the decreased abundance is simply because they have degrons in the protein that are then just causing degradation. Right? So these are real degrons, also in the sense that they work in the context of the full-length protein. The motifs and the sequence is relatively simple. So I have this wonderful picture of Christopher. He doesn't look exactly like this, but that's the picture of Christopher that's online. That's your fault, Christopher, and back to Tim's point earlier. And so we can train a very, very simple model. And actually, we tried more complicated model, but this is actually, you can do something relatively simple with a convolutional neural network. And we basically look at the internal stuff, and then we have a separate channel for the C-terminal stuff, and then we can predict abundance. So if we take just the internal channel and predict, or we chain and then we cross-validate, we get a high correlation, but then we have some outliers like this one here that has a C-terminal degron, and then if we add this channel from the C-terminal effect, we can put these back on the diagonal here. So we now have a model where you can apply this to any sequence that you might have if we're not covered by the tiles. Or for example, if you are interested in the effects of variance, so here's one, we analyzed a bunch of missense variants in exposed regions. Here's one example of a D2V variance in an exposed region that isn't a degron, but is a borderline degron in the wild type sequence. And then when you make that D into a V, you can see that you get a decreased abundance that you can then stabilize a little bit by bortezomib, but really showing that this, this missense variant, this pathogenic missense variant, is really, at least very likely, caused by, introducing a novel degron into the protein that then causes degradation simply without any unfolding. And so, clearly not all variants in disordered regions can be explained by this, but there's a statistical significant reason with these predicted changes in the abundance through Christopher's model. In the data from FIA, where we looked at transcription factors, we found that there is a strong overlap between transactivation domains and background, right, that they are both sort of hydrophobic. So here's a plot of, the transactivation domain as predicted by AD-PRED. This is the red curve, and the black curve here is the background predicted by Christopher's model. And you can see here that there's sort of a peak here in the region, and that's maybe not so surprising, but transactivation domains are hydrophobic because they sort of need to interact with mediate or whatever other things. So they are, sacrificing a little bit of being soluble because they need to interact. And if you look at this library again, if you just look at the tiles from the transcription factors that are predicted to be transactivation domains, we see that there are lower abundance than those that are not. If you look at the literature, there's a huge literature on the importance of hydrophobic residues and negatively charged residues in transcription factors, all the way back to this wonderful Paul Ziegler paper about the acid blobs. So we said, well, maybe one of the reasons why there's these negatively charged residues is simply to avoid degradation. So we can take this model, and we can just say we take a bunch of transactivation domains that have negative residue, we just do a single amino acid change. This is all computational. Not surprisingly, they all get they get stabilized a little bit. So maybe one of the reasons why there's negative residues in transcription factors, in particular close to the transactivation domains, is simply because they need to be hydrophobic, but they also need not to be degraded. And so we don't think that this explains all of it, but maybe it explains some fraction of the importance of this. And I'll just finish here with a really nice deep mutational scanning of a transcription factor, CRX. This is, work from Barrett Conan. So if you take this data set, this entire maybe to try to predict it with ESM, you get sort of a non-zero correlation in the DNA binding domain, but not a strong correlation in the transactivation or the disordered bit. But if you then take just this abundance predictor and you just, try to predict this map over here with the change in abundance, actually it's a pretty good predictor of this activity measurement over here. And so if you combine the two, you actually end up with a reasonable prediction. So you can explain, while I don't know whether this is causative, at least a substantial of the variation in this activity here simply by changes in abundance. And you can think that you can try to normalize your way out of this, and maybe you can, but I don't think that the activity of transcription factors is linear in the concentration of transcription factor. So I think just dividing by the amount of protein is not going to cut it if you want to normalize your way out of this. But the right way of doing this, I really don't know. So I know I've been speaking quickly, which means that I'm not so much behind on time. So thank you very much. We are very interested in continuing these kinds of things. Some things work fantastically well. Most things don't work so well. We're really happy that there's so much nice data being shared. Rasmus and my other colleagues is generating wonderful data. These are the people that did the work. I mean, the pioneer in the glucokinase work is really Sarah. Matteo did a lot of the work in the functional size together with Nicholas and others. Tia did the abundance analysis that I showed again with others, and then Basilius and Fia really did all the experiments of the difference, and I'm just happy to be sitting on the side. Thank you very much for your attention.